So, as I said, we are in transition from the digestive system, we're going into the urinary system, and today we're going to learn a little biochemistry. Just kidding, just a little bit, enough for you to be dangerous, but not actually know anything. So, on to redox reactions. What? This is an A&P class. I know, but you have to build from the ground up. So, ultimately, we're going to talk about how we metabolize lipids, carbohydrates, and proteins, why the liver is necessary for that, why the kidneys are necessary for that. That's where we're going. Why do we need our liver? Why do we need our kidney? We need to talk about this first, okay? So, this is chemistry, okay? There's a carbon in the middle here, and carbon has different redox states. And so the oxidative or reductive state of carbon depends on how many of the 4 binding sites has hydrogen in the fully reduced form here in the red. Okay, 1, 2, 3, 4, all bound to hydrogen, fully reduced. Okay, all the way to this side where 1, 2, 3, 4 binding sites are bound to oxygen, fully oxidized. What do we breathe out at the end of respiration? CO2, that's a waste product. So interestingly enough, we take in food and fuel as a solid, sucrose, glucose, okay, fats, and we breathe it out as a gas. Okay? CO2 is a gas, we breathe it out. But we eat it and then we breathe it out. So what do you do if you want to lose weight or whatever? You have to breathe a lot. Not really, but that's built into how we remove carbon from our body. We don't think of removing carbon, but fats, lipids, carbohydrates leave the body as a gas. We poop things out, but that's not how that works. It's not like we can hydrolyze fat and it goes into our feces. That doesn't work that way. Okay? We actually lose a lot of it through our breathing. Okay? So, reduced versus oxidized, these redox reactions are always paired. So in any reaction, if we have oxidation happening, okay, we're all still going to have a reduction happening. Okay? They always happen in pairs. And so in our human biology, the way that we utilize fuels is to take a carbon source like glucose, which is kind of somewhere in the middle of this oxidation state, or amino acids, which are also somewhere here in the middle, and then we fully oxidize that carbon. We oxidize the carbon and we reduce the oxygen, ironically. Fatty acids are more over here. So fatty acid chains are a bunch of saturated fatty acids. They have just a bunch of H's around them. They're really reduced. And so that's why the caloric content of fats is higher than the caloric content of sugars and proteins. Okay? I'm not going to tell you the numbers. I don't want you to memorize the numbers. You memorize that in your nutrition class. But the reason why fats get more calories out of them is because they're more reduced. And then we oxidize that fully, we get more calories out of it. That makes sense? Sugars and proteins are kind of in the middle here of the oxidative state for carbon. We can still strip some electrons away. So when we're moving from these H's to the O, I want you to think about ripping electrons off. Okay? Here, carbon's, I'm going to call it happy, he has lots of electrons. Psst, hydrogen carries electrons. Okay? Hydrogens carry electrons. So when we remove the electron, we remove that carbon's, when we remove the hydrogen, we remove the carbon's electrons. Here, oxygen is an electron hog. It's pulling all those electrons away, so carbon doesn't get to see any of it. Carbon's oxidized. So, for metabolism, we're taking a carbon source, some fuel, and we're oxidizing it. And we're taking oxygen, the gas that we breathe in and out, and we're reducing oxygen into water. Okay? Because we start with oxygen, oxygen, and then we end up with water, which is oxygen paired to hydrogen. Okay? So oxygen gets reduced, ironically. That's just that if you get through your biology degree and you don't understand redox reactions and how fuels work, we've not done a good job here at OU. So make sure you understand redox reactions. All right, so Again, this is pretty high level. I'm not going to ask you to memorize individual reactions. That's not the point of this, is to give you a sense of the overview of these metabolic pathways. Okay? So 3 fuel types: proteins, sugars, and fats. All 3 of these can be used to harvest and get ATP. The end result is make energy. Okay? This is all of a catabolic state. We're breaking things down and we're making energy. We also have anabolic state, but that's not what we're talking about right now. Okay, so we're going to focus here on glucose, and how glucose comes into these 2 and 3 carbon states. We go into the Krebs cycle and the electron transport chain, and then by analogy, we're going to follow proteins and lipids and how they feed into that same pathway. Okay, but we're going to focus first here on carbohydrate metabolism. We're going to take a couple notes on the board, but there's essentially 3 phases that you have to know, okay? 3 steps: glycolysis, Krebs cycle, electron transport chain. Okay? It says ETS here, ETC, electron transport chain or system, those are the same thing. Okay? 1, 2, 3. And lipids and proteins feed in this kind of in-between state that I'll talk about, the pyruvate or the acetyl-CoA. All of these steps, this figure is illustrating the cell. Okay, so most of these steps are happening in the cytoplasm. From Krebs cycle and ETS are happening in the mitochondria. That's important. You should know that. All right, so let's focus in here on glucose. Okay, glucose starts outside of the cell because we just digested it through our GI tract, right? We've broken down some piece of bread. We've had some amylase, cut it up into disaccharides, and then the disaccharide-ases have chopped that up, and we've absorbed glucose. Glucose gets imported into the cell, and we can begin the process of glycolysis. One of the first things that happens to glucose to lock it into the cell so that it doesn't zip back out of the cell is we phosphorylate it. Okay? A little P here is a phosphorus, okay, surrounded by oxygens. It's a phosphate. The important thing about glucose for our purposes is not that you've memorized the structure, okay? Which, by the way, looks something like this, which I'm not going to draw the structure. Either that's a glucose. Don't worry about drawing it, okay? That's just going to equal a circle. That's glucose. So we bring that into the cell and we put a phosphate on it, okay? And that phosphate actually locks it into the cell. Okay, that's kind of important. You'll need to know that for biochemistry. But for the rest of us who don't care about biochem, don't worry about that. There's a set of steps here, okay? What's important about glucose, as I was getting to say, was not to memorize this ring structure or whatever, but to know that there's how many carbons in glucose? 6. 6 carbons, okay? Let's count with our fingers. 1, 2, 3, 4, 5, 6. Love. 6. And they're kind of attached like this. 1, 2, 3, 4, 5, 6. So carbons here, right? At the end of this process, we're going to break that 6 carbon chain into 2. And we have pyruvate and pyruvate. Okay? That's where we're going. How do we do that? We need to weaken this middle bond. So what we can do is we can phosphorylate the ends of the glucose. We have phosphate here, and we're going to slap another phosphate over here. Those phosphates are electron hogs. They're going to suck the electrons away from the middle carbon here and weaken this bond a little bit and allow it to be easily broken into 2. Now we have 2 3-carbon molecules that will eventually we're going to call pyruvate. Okay, so we go for 6 carbon chain, glucose. We add some phosphates to kind of manipulate the electron cloud a little bit. And now we can break that bugger in half. Okay? Because this down lower is the substrate that we need for glycolysis. So these are the rest of the steps. There's 8 to 10 steps, depending on if you count the other side of the pathway. So there's lots of steps, lots of enzymes. Don't worry about memorizing those. Okay? What I want you to know is glucose and pyruvate. Okay? We start with glucose. How many carbons? 6. You end up with 2 pyruvates. How many carbons of pyruvate? 3. Okay, we've just broken it in half. Now, just like a good investment, okay, I'm not a money guy. My wife handles all of our funds. She does a fantastic job. I'm thankful. Proverbs 31, all that, okay? Seriously, guys, if you're not a money guy, find a woman, handle the budget. Anyways, investment. I don't know how I get these tangents. It takes money to make money, right? You have to put a little money into something to get money out of it. And that's true with glycolysis. So the first 2 steps, we need to spend to ATP, because we need to phosphorylate the ends of the glucose. So we're going to take a little bit of investment. We just spend 2 ATP, it's -2. But at the end of this process, we're going to gain 4. So 4 plus 4 minus 2 equals a net of 2 ATP. Every time we go through glycolysis, we gain a +2 net ATP. We make 4, but we spent 2. So net of 2. You need to know that. Okay. All of this does not require oxygen. Okay? It is anaerobic. Okay? So this is a good way to get energy any way, any condition, but oxygen or not oxygen. And we end up with these 2 molecules that are 3 carbons of pyruvate. From pyruvate, okay, let's start to write this down on the board over here, just to have something down on the board. Carbohydrate metabolism. At the top, we're going to start with glucose. Glucose is a 6-carbon molecule. And in this process called glycolysis, there are about 10 steps, 10 biochemical steps of processing this, and all the steps are important, all the enzymes are important, but not for our class. We end up with 2 pyruvates. And these are 3 carbon molecules. Okay, that's glycolysis. And there's actually a branch here. Okay, it doesn't matter. Don't just forget I drew that. But there's actually the splitting, and you have to kind of convert it back in, whatever. From pyruvate, we turn this into another molecule called acetyl-CoA. Acetyl-CoA. There's 2 of them, and this is a 2-carbon molecule. That's important. Where did the other carbon go? Well, we lost it as carbon dioxide. Okay, there's a redox reaction here. We'll talk about where those electrons went. But we lost a carbon because we made some carbon dioxide. This step is kind of an in-betweener step. It's in between glycolysis and what comes next, which is the Krebs cycle. Acetyl-CoA is the entering substrate for Krebs cycle. Okay, so let's talk about that here. This enters in, we go around the TCA, or the Krebs, K-R-E-B, cycle. TCA, the tricarboxylic acid cycle. The Krebs cycle is another series of biochemical reactions. There's about 10 reactions. They all have a different enzyme, they all have different individual intermediate substrates, and coming out of the Krebs cycle are a number of molecules that are important. So let's go back to the slides here on the board.
Why is it called the citric acid cycle where there's no citric acid in it at all?
That is, I don't have an immediate answer. If I looked at this carefully, I could probably figure that out why that is. This is citrate here. So citrate, citric acid, one is the conjugate, so I think that's the reason.
I just call it.
My PhD is in biochemistry, but I'm not really a biochemist. I'm more of a genomics guy, which is my PhD was on. I hope nobody from my department hears me say that. So this is the citric acid cycle, entering substrate, acetyl-CoA. How many carbons is that? 2, okay? At this step, we went from pyruvate to acetyl-CoA, we lost a carbon. And we went from 3 carbons to 2 carbons, okay, waste product. Okay, what do we breathe out at the end of respiration? CO2, okay, it's a waste product. We've also made, when we lose CO2, typically we're also making an NADH, NAD plus to NADH. So let's talk about that just a minute. NADH, 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 FADH2, what's that? It's kind of similar, so think about them as cousins, NADH. So NADH, NAD plus, is a coenzyme. It's an electron carrier. Okay? It's an electron carrier. All of these individual steps, not all of them, but many of them, have a process where they're doing a redox reaction, and they're stripping away the electrons from the carbon, and they're giving them to these carriers. They're carrying electrons to the electron transport chain. Okay? That's their purpose. I used the analogy this morning. It just comes to my head and I blurted it out before actually kind of thinking about it. It's like when you're at the casino. I've never been to a casino, ever. I'm not into gambling, don't promote it. We have the chips, right? You win the chips. The chips themselves don't have any monetary value until you cash them in. Then they're actually worth something. But in the casino, they're worth something, but outside the casino, they're not worth anything. Anyways, FADH and NADH are like the chips. They're not worth anything until you go to electron transport chain and you turn them in and you get the ATP. ATP is worth something, okay? So they carry the electrons to the electron transport system. And then we drop them off and we do oxidative phosphorylation. Along the way, we're making some CO2 as waste, and because at the end of this we end up with oxaloacetate, which is a 4-carbon molecule. And we add this 2 carbon, and we end up with another 6 carbon molecule. And you don't have to know that, but the math works out. One of these steps, we actually make an ATP directly, except it's not ATP, it's GTP, but for our purposes, it's the same thing. So GTP is made, but it's a direct substrate level phosphorylation, and we get ATP there. I will go through the numbers, okay, the 32 versus the 36 ATP at the end, so don't worry about that right now. This is the more detailed version of that same picture. You can see all the different substrates, all of the enzymes in there are involved. You do not have to memorize those, so don't sweat it. What you do have to know is that there's NADH coming out, there's CO2 leaving as waste, and in one step here, we're actually making GTP, which we can make ATP. So let's write that down over here. Coming out of the TCA cycle, we have NADH. We're also having FADH2, and we're actually having directly ATP. I know the folks in the back can't read that. I'll just read them again loudly so you can understand. Coming out of the TCA cycle, we have NADH, a bunch of those. We have FADH2, and we have ATP. All of these will feed into the different color, the electron transport chain. Okay? The electron transport chain. We're carrying electrons into the electron transport chain, and out of that we get ATP. That's the whole purpose of this. So, as we're kicking around these individual reactions, we're generating NADH. Usually the steps combined with the CO2 kick out. Okay, NADH, CO2, NADH, CO2. Okay, and here this is FADH2. It's a similar type of thing. It's carrying electrons. Okay, all of these will feed into the electron transport chain. So this is what that would look like in the biochemical schematic. Okay, NADH or FADH are feeding into a series of protein enzymes, CoQ, cytochrome B, C, A, and A3. Don't have to memorize them, but they're sitting inside of the membrane of the mitochondria. Okay, CoQ, A, B, C, cytochrome. All of these, if you follow this red arrow, the electrons are passing through a series of reactions. The end result is to power the ATP synthase, okay? This is like a big rotor that rotates. And when it rotates, it phosphorylates ADP into ATP, okay? What does the D in ADP stand for? Di, which means how many? 2. Tri means 3. So we're adding one phosphate from ADP to ATP. Simple, but very important. This is the machine that makes ATP. We need to power the machine by electrons. Really simplifying this, but you're going to memorize this, and you'll have it the rest of your life. Or at least till the next exam. All right? We're making ATP. We're also making water. Okay, because part of the process here is making water. All of these steps serve to pump protons into this space, and as the protons build up a concentration gradient, they pass through this, they turn the crank, and make ATP. That's all I need. Very simple. All right. So, overview. We started off with a 6-carbon glucose. Go through glycolysis, we end up with 3 carbons, and then eventually 2 carbons. We go through the TCA cycle twice because we have 2 pyruvates and 2 acetyl-CoA, so we have to go through this twice, okay, per 1 glucose. We generate some electron carriers, which will carry the electrons into the electron transport chain with this chain of reactions where they're tumbling down the redox states, and that's going to help us to make ATP. And along the way, we're also making a whole bunch of CO2. Remember, that C of CO2 is the most oxidized form of glucose. And we've broken a bit off of it, we've oxidized it, and we're getting rid of it as respiratory waste. Now, all of this flow through this pathway assumes demand. We need ATP, right? We need energy. All the time, constantly, right? Not really. Sometimes the cells, just like we're chill, we got plenty of energy, and we got way too much glucose. We just had like 4 Krispy Kremes or whatever, okay? Lots of glucose, and we have excess glucose, so what are we gonna do with that glucose? Just sitting around doing nothing. We're gonna make glycogen, very good. Okay, so here's the pathway, here's glucose. We can go this direction, glycolysis, and feed it to Krebs cycle. Okay, that's what we just talked about, from here down. Glycolysis. Okay, let's go over here. When you think of the word lysis, what do you think? What does lysis mean?
Cut apart.
To what?
Cut apart.
To cut apart, to break. Lysis means to break. If you have hemolytic disease or hemolysis, means your red blood cells are breaking apart. A lytic or lysis. Genesis means what? Creation. To build, to make, right? So these are opposite, these are antonyms in some sense. And we're going to learn that glucose and glycogen are important concepts. Let's go over here back to my monomer. This is one molecule, one glucose. If we put those into a chain, very simplistic drawing here, we get a polymer which we call glycogen. Okay? It's just a polymer of glucose. We've made a covalent bond. It's a 1,4 alpha glycolytic bond. You don't have to know that, but that's what it is. Alpha is easily digestible, okay? Not beta. That's the storage form of glucose. Okay? What are the 2 places in our body that store glycogen? Muscles and liver. Okay? We store it in our muscles, we store it in the liver. Okay? If you're athletes and you carb load, eat a bunch of like rice or pasta the day before your big game or your running event, you're carb loading, meaning you're loading up your liver and you're loading up your muscle with glycogen. Okay? Glycogen is this storage form. Okay? And it's easily accessible in the liver. Our bodies are built centered around glucose metabolism. it literally looks around the whole body, and if it sees glucose, we're gonna burn that first. And if all of that glucose is burned up, including all of the glycogen, well then we might decide to burn a little fat or protein. And that's why it's so darn hard to lose weight if we're desire to do that. Okay, because it will take any sugars and do that first. Then you have to kind of deplete that and then go into fatty acid oxidation or other kind of ketoacidosis, burning our amino acids. Anyway, so our body prefers glucose, and so restricting glucose is a way of really changing body formation. This process on this side is called glycolysis. We're lysing, we're breaking apart glyco, which is glucose. Okay. Now, a couple of these other arrows that we need to learn about. 1, 2, 3, 4 that we're going to learn about. Glycolysis is the one that we talked about. When we go from glucose and we make glycogen, it's called glycogenesis. Glycogenesis. So we're making glycogen. We're forming glycogen. So if you know the root words, it becomes super easy. Okay? Glycogenesis. We're taking glucose and we're forming that into glycogen. Which organs does this happen in? Liver and muscle, primarily. Okay? Glycogenesis happens in liver and muscle. If we're taking glycogen and let's say we've been lazy and sitting on the couch for 3 days and eating our bonbons, okay? We've stored up a whole bunch of glycogen and we're just kind of storing that. Now, on that third day, we've decided to go run a 10K, okay? Why would we do that? That's silly. But we're gonna do that, so we need to break down some of that glycogen into glucose so we can make ATP and fuel our muscles. Because Mr. Myosin Head needs to hydrolyze ATP to pull on the muscle on the actin. Okay, so we need energy, there's a demand, so we're going to break down glycogen into glucose. We call that glycogenolysis. Glycogenolysis, okay? We're breaking down glycogen. And in the process, we need glucose, which is going to feed right into glycolysis, we're going to make ATP. So, this direction. So those are pretty easy. This one's kind of a confusing one. Okay, this one's called, let's see if we can work it out. Gluconeogenesis. Neo means new. Genesis means form. So it's the new formation of glucose. New formation. So actually it's this pathway from these 2 and 3 carbon precursors going up and forming glucose this direction. This happens in a fasting state. We're plumb out of glycogen, so we can't form glucose that way. We can form glucose through this other unique pathway from other substrates from fatty acids or amino acids. It's called gluconeogenesis, forming glucose from the bottom up. It's a weirdo, it doesn't happen frequently, but the pathways do exist. All right. As we reverse our way through glycolysis, there's a couple detours that we have to make because the reverse pathway is energetically disfavorable. But again, you don't have to worry about that. In biochem, you'll worry about that if you can ever change it. That's carbohydrate metabolism. Any questions on any of that right now? Okay, this is just big picture stuff. Now, let's talk a little bit about lipids. That's a squeaker. Lipids. And we said that lipids have a higher caloric content than sugars because they have more reduced carbons. Here is Mr. Triglyceride, okay? Fatty acid chain 1, fatty acid chain 2, fatty acid chain 3, 1, 2, 3, triglyceride, okay? This molecule that holds them together is called glycerol. And these chains can go through a process called beta oxidation. Beta oxidation. Very simplistic terms, you're taking the end chain and we're breaking off 2 carbons, and 2 carbons, 2 carbons, we're breaking them off, okay? Cut. We're using those 2 carbons, where could we fit in 2 carbons into this system? Acetyl-CoA, okay? So we go through the process, lipids called beta oxidation. And we can fit those substrates right in there and run those buggers right through a TCA cycle, electron transport chain. The glycerol can fit in at the 3-carbon stage, and because it's 3 carbons, the beta oxidation would break off 2 carbons at a time and feed it right into acetyl-CoA. There's way more in here, but we're not going to learn it. That's as much as we need to know for today. All right, this is how it gets absorbed. We've talked about that a little bit already from the lacteals, so let's split it up. Now, lipids was easy, okay? Proteins, another deal. The colors don't really mean anything, they're just different. So this is glucose, but this is going to be protein. Proteins, okay? Lipids, glucose, proteins. The third of the nutritive molecules. What element does protein have that lipids and carbs do not have? Nitrogen. Okay, proteins have nitrogen. Let's do a little drawing here, just to remind yourselves of the general structure of amino acids. Okay, we have an amine group, we have the central carbon, and we have the carboxylic acid. Okay, this is called the alpha carbon, that's where the R group is, and there's some chain hanging off, and this is just going to be amino acids. This is the main chain of every single amino acid. NCC, 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 okay? And on. And then sticking off the side is the R group, or the variable group. Okay? And that could be as simple as an H, could be as complex as a proline, and lots of other side chains. And these have its own metabolism that we're not going to talk about. But every single amino acid has a nitrogen and 2 carbons. Okay? These 2 carbons, I wonder where we could fit 2 carbons into this. Okay? So here's a protein, you have an NCC backbone. These 2 carbons are going to fit right into here. Right below acetyl-CoA. We can utilize those carbons. They are at some level of oxidation. We can still strip more electrons off them, and we can use them for Krebs cycle. Oh, wait a second. What about, use a marker here. What about this guy? That's the problem child. That's a nitrogen. That is, if we break that off, we call that ammonia. Ammonia is toxic to us. Ammonia is toxic to the brain. Okay? And if it accumulates in the blood, we got problems. So every time we break down proteins, we gotta figure out what are we gonna do with that nasty nitrogen. And it's not easily excretable. We can't get rid of straight up ammonia. So there's a couple processes that we need to use. Okay? Here's glutamic acid. Here is the NCC, they have to put them in a straight chain. Here's the side chain, NCC. Transamination and deamination are ways of breaking off the ammonia. Okay? So we're going to take this regular old amino acid, break off the ammonia, and now that's the part we need to get rid of. So we have this beautiful cycle called the urea cycle. In the urea cycle, we have 2 ammonias. Ammonium is the ion, 4+, so there's an extra proton on there. Don't get confused, it just has an extra hydrogen. Ammonium is the ion, ammonia is NH3. We take 2 of those, add a carbon dioxide, go through a series of about 5 reactions using ornithine and citrulline and a whole bunch of stuff in the liver, and we make urea. Here's that one nitrogen, here's the other one, and here's our CO2. And we just added them together. It's not quite that easy, there's a set of steps involved, but we don't have to know those steps. Urea cycle happens in which organ?
The kidneys.
The liver, okay? The liver. The urea cycle happens in the liver. What's the input to the urea cycle? Ammonia and CO2. Where does it happen? The liver. What do we get out of it? Urea. Right? Urea comes out in the blood. Where does urea go in the blood? To the kidney. Okay? And the kidney says, I know you, you're leaving in the urine. Okay? So it pulls it out of the blood, and the kidney puts it into the urine, and we pee it out. Yay, successful, we've gotten rid of the problem child, nitrogen. Very important because we need to get rid of urea. We've gotten rid of 2 of them here in one shebang. There are other nitrogenous wastes that we have to be aware of. Creatinine forms from creatine, that's a muscle breakdown product, also chock full of nitrogen, also pulled out by the kidneys. We're going to get into that on Friday, right? Well, probably next week. Friday will be mostly anatomy. Tell me what time is it? 10:20. Okay. A couple more key concepts and we'll be done. Alrighty. So hopefully we've overviewed how do we understand carbohydrate metabolism. 3 major phases. Glycolysis. Okay, glycolysis, 1. Krebs cycle, 2. Electron transport chain, 3. How do lipids and proteins fit into that for making energy? Well, lipids, there's a complicated process whereby we can break down glycerol and those 2 carbons. We can fit them in right here at the 2 carbon level. Also proteins, because the individual subunits here have 2 carbons, we can fit them in here at acetyl-CoA as well, and then go through electron transport chain. Proteins are special because they have nitrogen, so we have to get rid of those ammonia, and we have to use the urea cycle to do that. At the end of electron transport chain, we can get up to 36 ATP from 1 glucose. Okay? 36 ATP. It depends on how many NAD and FADs there are, sometimes 32, sometimes 36. It's variable and confusing, but that's biology, so memorize 36. That primes us, you, for going into the urinary system on Friday. Okay? Thank you. See you guys Friday. All of these slides will be put on Moodle, okay?